Today we will be looking at selection and speciation. Our learning objectives for today are to explain the process of evolution and to explain the process of speciation. Both of these are going to be refreshers from first years. And finally, we'll be exploring the molecular evidence that can be used for evolution. Now, when you are writing an evolution answer, there are certain things you should always include, and so you should learn this wording. Firstly, random mutations produce new alleles. We then need to mention that there has been a change in the environment or there has been a selection pressure. This will differ depending on what type of exam question you get. We always want to relate it back to the scenario given. Now, some of these organisms in these new selection pressures, in these new environments, will now be better adapted for their environment because they have the right mutations. Mutations are always happening. It doesn't really matter what the environment is for mutations to happen. It's just that some of them will actually help the organism be more adapted for their environment. Those organisms are then more likely to survive and reproduce. Again, those are key terms there. Survive and reproduce to pass on their alleles to their offspring. Those that have not got those alleles will be less adapted, and so they'll be more likely to die from predation, from competition, and from disease. Over time, those that did have the advantageous alleles will keep on passing on those alleles to further offspring, and that means that over time, the proportion of individuals with the favorable genotype will increase. At the end of all this, we say we've had a change in the allele frequency. And I'll show you this using an example. Let's say we've got a population of reindeer. Now, all the reindeer look different. There are random mutations. Some of them have quite short fur, some of them have quite long fur. Now, let's assume that a selection pressure is available. We've gone from quite cold temperatures in 1810 to warmer temperatures in 1960. Some of the organisms, some of the reindeer, have shorter fur because of that random mutation, and so they just happen to be better adapted for this new environment. Those are then more likely to survive and reproduce, whereas those that are less adapted, less well adapted, will die. Over time, the proportion of individuals with the favorable alleles will increase. So we keep getting offspring with quite short fur because those were the parents that actually were able to survive and reproduce because of their short fur. And then over time, a larger proportion of individuals of the population will have the advantageous alleles, and so will have short fur. Okay, this evolution answer that I've just presented to you has to be applied to the scenario in the question. You can't just regurgitate a standard answer because you will still lose marks. So, as a way of trying that, we can attempt an exam question. This will be available to you on Moodle. So, treating Mycobacterium tuberculosis infections can be a problem, as the bacteria are resistant to many antibiotics. There are many strains of Mycobacterium tuberculosis. Different strains are resistant to different antibiotics or combinations of antibiotics. The chart below shows the percentage of resistant strains of Mycobacterium tuberculosis to 6 different antibiotics or combinations of antibiotics in 2006, 2007, and 2008. And we can see that. So the first thing we want to do is try to understand what the graph is showing us. We've got the years here on the x-axis, and then we've got percentage of resistant strains of Mycobacterium tuberculosis. In the key here, we've got the different types of antibiotics. Sometimes they're by themselves, like streptomycin, ethambutol, but then some of them are combinations. So, for example, INH and rifampicin. So, when we look at this graph, we can see that in 2006, about 3% of Mycobacterium tuberculosis were resistant to INH. Okay? And it seems like over time that resistance just increased. So more and more of the bacterial population was actually getting resistant to this particular antibiotic. Now, first question. Using the information in the graph, compare the types of antibiotics and combinations of antibiotics that the Mycobacterium tuberculosis are resistant to in 2006 and 2007. Okay, so that means we can ignore 2008 completely. For 3 marks, all you need to do is try to compare what's happening in 2006 and what's happening in 2007. Have a look at which antibiotics or antibiotic combinations these bacteria are resistant to. So if you stop the video now and try to answer it, and then after that I will go through the answer. Okay, hopefully you have now answered that 3 marker. So remember, we are ignoring 2008 because that's not what the question is asking us about. So the first mark is available. The idea that bacteria are resistant to fewer antibiotics or antibiotic combinations in 2006 than in 2007. And that makes sense. You can see that in 2006 the bacteria are resistant to 1, 2, 3, 4, whereas in 2007 it's 1, 2, 3, 4, 5. Okay, you would have also gotten that mark if you describe it. So there are 4 antibiotics or antibiotic combinations that the bacteria is resistant to in the first year, and it increases to 5 in 2007, the second year. In both years, there are resistant strains to streptomycin, and if we have a look at that, yeah, that makes sense. There's streptomycin here, streptomycin resistance here as well. Both are resistant to INH, rifampicin, and ethambutol. Right here, yep, both are resistant. And finally, INH, which again, we see that here. Any one of those would have gotten you that mark. Number 3. The idea that there are resistant strains to INH and rifampicin in 2006 but not 2007. So if we look at that, this is not the darkest grey but quite a dark grey. Bacteria are resistant to this in 2006 but that disappears completely in 2007. And final mark, the idea that there are resistant strains to ethambutol rifampicin in 2007 but not 2006. Which again, that makes sense because we have this really dark grey colour for this antibiotic here, and that only starts appearing in 2007. We did not see that in 2006. Okay, so that's 3 marks available there. Now, second question. The percentage of strains of Mycobacterium tuberculosis resistant to the antibiotic INH has increased during the 3 years. Can we see that there? Yes, we can. That was the white bar there. Suggest how natural selection would have resulted in this increase. Have a go at answering this question. Stop the video now. Okay, we'll go through the answer then. Now, whenever we talk about natural selection and evolution, we should always start with the random mutation. So, the bacteria must have acquired a mutation in their DNA or their genes. You get another mark for the idea that the presence of an antibiotic is the selection pressure. In the reindeer question, the weather or the temperature was the selection pressure. In this case, it's whether there is an antibiotic present. The idea that the allele for resistance gets passed on between the bacterial generations. Notice that you cannot say gene, it has to be the allele. Now, you do get a mark if you mention how bacteria reproduce, because it's different for them. And this is the mark that gives you, for this mark you need to say that they divide by asexual reproduction or binary fission. This is the most correct answer. You would have also gotten that mark if you say that it's mitosis or conjugation or transduction or transformation. These are 3 different ways of bacteria passing on information between each other. The idea of increasing allele frequency, and the idea that the more resistant bacteria there are, the more likely that new strains will acquire their resistance. Okay, so we're looking at the idea that there is an antibiotic there, that's the selection pressure, but some bacteria have a mutation in their DNA that they can pass on to their offspring when they reproduce by binary fission, and over time that includes the allele frequency. Okay, any of those would have gotten you a mark, and the 3 marks is the most you can get. Final question. Suggest how hospitals could prevent an increase in the percentage of strains of Mycobacterium tuberculosis resistant to antibiotics. Okay, so this is a slightly different question now. It's not asking us about evolution, and it's not asking us to describe a graph now. So if you stop the video here and try to get your 2 marks, and then now we'll go through. So, reference to codes of practice or codes of conduct. So if you said specific things that you know hospitals are doing, you would have gotten this mark. Now we'll look at the rest of the answer first before we give examples. The idea that appropriate antibiotics should be given to patients. This is the same as saying that you should not be giving antibiotics to patients if they are not necessary, or you should not be giving antibiotics to try to prevent disease. You should be using antibiotics only when they are absolutely necessary and for the correct infection. The idea of educating patients about taking antibiotics. We know that there's quite a few people out there who still take antibiotics until they feel better and not actually for the full course that's required. And that means that actually some of the hardiest bacteria are actually able to increase in number and start causing disease again. Credit another appropriate procedure, for example, hand washing, screening, changing beds of patients more frequently, separating patients into different sections of the hospital if they have infectious diseases. Any one of those would have been correct. Okay, so that was evolution. Now we're going to have a quick refresher of speciation, which you will have covered a little bit of at AS already. So speciation is generally species forming because there has been something that has stopped them from being able to reproduce together. So they might have a common ancestor, or they will have a common ancestor, but something has happened where we have formed new species. And the official definition is, speciation is the formation of new species by populations that have been reproductively isolated. Key term there, reproductively isolated. Now, there is a few different ways in which populations can become reproductively isolated, and those are ecological isolation. It could be that the species are located in the same area, but they occupy different parts of the habitat. So it could be that you have species of bird, and one of the species is always found in the top of the trees, whereas the other species tends to occupy lower branches of the trees, and so they never overlap. It could be temporal isolation. Species exist in the same area, but they reproduce at different times. This could be something like frogs. Some of them may do their courting calls right in the morning, whereas the others do it late at night, so they never actually encounter each other. Behavioral isolation. Some species, or the species exist in the same area, but they do not respond to each other's courtship behavior. So there might have been a slight change. If this was a bird, the dance might look different, or the call might look different, and so they are not attracted to each other. Then we get into physical incompatibility, and that's where species again live in the same area, but there are physical reasons that prevent them from copulating. This could be something like if you think about Chihuahuas and Great Danes, those are still the same species, but there would be physical reasons that prevent them from being able to copulate. Although they are still the same species, it could be that if we keep selectively breeding them for their looks, eventually, maybe over thousands and thousands of years, if not millions, they are to become a separate species. Then we have hybrid inviability, and that's when we can produce a hybrid. So the two species do breed, but the hybrids themselves, so the offspring, cannot themselves survive long enough to breed. And finally, hybrid sterility, which is when, again, two species form a hybrid, but they cannot reproduce. Classic example is having a horse and a donkey. They produce the hybrid, which is a mule, and the mule cannot reproduce to have their own offspring. Now, there are two types of speciation. It depends on how the populations have been isolated. The first type is allopatric speciation, and that's where you have some sort of geographical barrier that prevents organisms from interbreeding. And finally, sympatric speciation, which is where the species still share the same habitat, but there has been some other reason as to why they are not interbreeding together. And that could be something like time of mating, it could be something like the courtship changes, it could be something like, from our other examples, mating at different times. It could be that they are just in different parts of the same habitat. Now, we start off with the common ancestor, and let's imagine that in our population, half of them have the selection pressure of there being a warmer temperature, and half of them have the selection pressure of there being colder temperature. Because those are different selection pressures, allele frequencies will change according to the conditions X in this particular population. Okay, so the random mutations are always happening, but it just so happens that to survive on this side, it makes sense to have alleles that help you survive the warmer temperatures. On this side, it makes sense to have alleles that help you survive colder temperatures, and so the allele frequency changes according to those conditions. Over time, that results in two different populations. If enough time has passed, they would start to look different, and if you tried to get them to produce fertile offspring, if they cannot do that, then you know that this is a new species. Just like we saw with evolution, there is also a exam answer for speciation, and we always start with populations getting separated. Now that could be allopatric separation or sympatric. This separation causes them to become reproductively isolated. That means they cannot interbreed together. That's a third mark. If they cannot interbreed, the gene flow between the populations is reduced. That means that each population then accumulates different random mutations, and so their gene pools are starting to have different alleles. You could have also gotten that mark if you say that the variation in small isolated populations may be greatly reduced because some alleles may be lost to genetic drift. So something like diseases, or something like physical catastrophes like fires can kill a lot of the population, and that means that the alleles left are quite limited. You could have also said that genetic diversity may be reduced because of the inbreeding depression. So there's a very small population there, and so you keep getting more and more of the same alleles, especially if they're recessive alleles, and that can result in a lot of diseases showing up in the phenotype. 6. The isolated and the original population may now show significant differences. Each population has been subject to different selection pressures. This changes the frequencies of particular alleles in the gene pool and results in different gene pools. Individuals in each population have changed so much that they can no longer interbreed successfully. Now, to check whether you can understand that, we're going to have a go at another question. An experiment was conducted with fruit flies. One population was split into two, and each was fed on different food. After many generations, the two populations were placed together, and it was observed that they were unable to breed together. The questions I would like you to answer are: what evidence shows that speciation has occurred? Explain why the experiment resulted in speciation, and suggest two possible reasons why members of the two populations were not able to breed together. If you stop the video now and have a go. And now we can go through the answers. So firstly, what evidence shows that the speciation has occurred? Well, the new species were unable to breed with each other, or they were unable to produce fertile offspring. Number 2, explain why the experiment resulted in speciation. Well, populations were isolated and fed on different foods, different selection pressures. This caused changes to the allele frequencies in different parts of the populations. This made them reproductively isolated and eventually resulted in speciation. Final question: suggest two possible reasons why members of the two populations were not able to breed together. And if you mentioned any one of those, you would have gotten a mark. So seasonal changes, they may be breeding during different times. It could be mechanical changes, so they're physically not able to breed together anymore. It could be behavioral changes, so maybe their courtship behaviors have now changed. And finally, we can start looking at molecular evidence for evolution. So, all organisms on the planet contain a similar, very, very similar structure of DNA. This and protein synthesis are very similar in every organism on Earth. And that supports Darwin's idea of all living organisms sharing a common ancestor. Now, this image here is actually Darwin's drawing. So he has drawn a tree that shows that everything that lives on the planet has come from one single common ancestor, and this is how we got loads of different species. Now, if we look at the DNA and proteins, we can see how there are changes within that as we move through time because of random mutations, and we can actually use that to figure out how closely related different species are. So, there are two major areas of research happening here. One of those is called proteomics, and that's using the differences in amino acid sequences to determine how different species are. And there's also genomics, and that's looking at the differences in bases of DNA to look how different species are. Interestingly, there are certain genes and proteins that are conserved in all species. That means that they must be pretty important for something, and we call them housekeeping genes. So, genomics, the study of DNA, we look at the sequence of bases in genes. The more distantly related two species are, the more differences there will be between them because they have accumulated mutations over time. And we can look at that by looking at DNA sequencing, DNA hybridization, and DNA molecular clocks. So in DNA sequencing, you can use restriction enzymes to cut DNA into different sizes, and then you can use electrophoresis to separate them out based on size. And so you can see here that there was a fragment here. The shortest fragment would have been just one base, and that's cytosine. The second fragment would have had adenine as well. And you work your way through that to figure out the sequence of nucleotide bases within the DNA. Then you can put those things together and try to look how similar different organisms are. This particular example looks at the giant squid and the octopus. And wherever there is a similarity in their bases, they get a star, and where there's a difference, they don't get a star. And you can see that the majority of the DNA is quite similar between the giant squid and the octopus. But obviously, because they are different species, the DNA is not going to be exactly the same. Now, in DNA hybridization, what we do is when you heat DNA, it separates out into its component strands because the hydrogen bonds between the bases start to break. What you can then do is mix the strands of two different species to try to make hybrid DNA. Now, because not all of their base sequences are the same, not every base will be able to complementary base pair. And you can see here that there are certain bases that were not able to pair up. But the more complementary base pairs there are, the more closely related the species are. Now, in DNA hybridization, this is roughly, but a 1 degree rise in temperature corresponds to a 1% difference in DNA. So the more energy, the more temperature, the higher the temperature you need to separate out two strands, the more similar it means that the DNA is, because you're having to break more of the hydrogen bonds. If we were to look at that in more detail, let's say we've got species A, we've got species B, and we've got a species C. What we would do is we want to see how closely related those different species are. And so in our DNA hybridization experiments, we would separate the DNA of all of these and then mix species A and species B DNA together, species A and species C DNA together, and finally species B and species C DNA together to form hybrids. Now, you remember that adenine and thymine, the number of hydrogen bonds between them is 2. For cytosine and guanine, the number of hydrogen bonds between them is 3. If we were to look at this step by step and try to see where the bases match up, we would see something like this. Okay, so adenine and thymine bond together. Adenine and adenine are the same base, but they are not complementary to each other. Okay, so I've done that along the whole length of this DNA, and if I was to count the individual hydrogen bases, hydrogen bonds, we would get 28. For the second one, for the second hybrid, this is A to C, 25. Okay, so even the first base is different now. And then finally, C to B, we would have 20. Okay, so the more closely related the species are, the more complementary base pairs there are, the more hydrogen bonds there are, and so the higher the temperature you would need to separate those DNA fragments out. Now, I'm going to get you to have a go at this question. So to illustrate how to work out evolutionary relationships using DNA sequences, you'll be using the example of DNA sequences for 4 organisms in Table 1. For each pair of organisms, count and record the number of differences between their DNA in a grid-like pattern shown in Table 2. Okay, so you'll be going through and seeing how similar is A to B, how similar is A to C, how similar is A to D, and so on and so forth. Now, the top of the table here and the bottom of the table here are similar. So, for example, if you do B and A here, then you no longer need to do the B and A here. So actually, you can ignore this side of the table. All you need to do is fill out these boxes here. Now I'll do the first one with you. So we're looking at A and B. So that's a similarity there. And we're looking for differences. So let's look for differences. Okay, that's a difference there. That's a difference there. That's a difference there. There's a missing base. There's a difference there, difference there, difference there. That's a difference and that's a difference. So that's what 1, 2, 3. If I made a mistake somewhere, 1, 2, 3, 4, 5, 6, 7. Ignore that C there, sorry. There's 7 differences between A and B. If you stop the video now and try to fill out the rest of the table. Okay, and I'm going to show you the results. So you should have found that the most differences were found between the DNA of organism B and D. That was 11 differences there, and that means that B and D are the most distantly related organisms. The most closely related was A and C. There was only 1 difference in their DNA. Final bit of genomics is DNA molecular clocks. Now you can actually analyze DNA sequences to try to figure out how long ago did we have a common ancestor with different organisms. Now we know that over the course of millions of years, mutations accumulate in DNA and different genes mutate at different rates, which are actually quite reliable. So we're going to look at a specific example here. When a stretch of DNA does indeed behave like a molecular clock, it becomes a powerful tool for estimating the dates of lineage splitting events, and that's when we get that speciation. For example, imagine that a length of DNA found in 2 species differs by 4 bases, and we know that this entire length of DNA changes at a rate of approximately 1 base every 25 million years. That means that if we have 2 DNA versions and they differ by 100 million years of evolution, that their common ancestor lived 50 million years ago. Now I'll show you this as a diagram. Let's imagine that 50 million years ago we had a common ancestor, and this was their DNA sequence here that we're focusing on. After 25 million years, the 2 descendant lineages have diverged. Each of them has had a single mutation. So here we've gone from T to G, and here we've gone from that G to a T. Okay, so there's 1 base mutation every 25 million years, which is what happened for this lineage, and it's also happened for this lineage there. After another 25 million years, so overall that's 50 now, the 2 descendant lineages have diverged even further because, as we saw before, every 25 million years we get a base mutation, and so now we've got an additional C there instead of a T, and on this one we've gone from a C to another T. That means that these lineages now differ by 100 million years of evolution, because you go through 50 million here and then 50 million here. So it's not only just 50 million, because they have diverged into two and they have gone down their separate paths for 50 million years. This is what they actually used to try to figure out how long ago modern humans emerged, and how long ago we shared a common ancestor with chimps. Okay, that was done using molecular clocks for certain genes. Now finally, proteomics is a very similar concept to comparing DNA sequences, except this time we will be looking at seeing the differences in amino acid sequences. And again, the more distantly related two species are, the more differences you will see in their amino acid sequences. So if we were to look at this example, and we're comparing the proteins for a bunch of different animals, you can see here that mice and rats share quite a lot of their DNA, and that means that a lot of similarities will be seen in their amino acids as well. Now, if we compare that to a human, you can start seeing that actually for this particular stretch, or this particular protein, we've got the last two amino acids that are different. Okay, and that will differ depending on what stretch of DNA you have chosen. In this particular one, all of these organisms have the same first amino acids. There is a slight difference in the second one because a chicken has a different one. But if you were to analyze that for similarities, you can try to figure out how closely related these are. Now, all of this is done using bioinformatics, which is a really quite fast emerging branch of biology. And that's where scientists and researchers align primary structures of proteins to analyze them using computers, so that you don't have to sit there and try to figure out the exact similarities and differences. You can get a computer to do that for you. Now, all of this information still is subject to validation. Scientists cannot just publish this sort of stuff and hope for the best. It does have to go through quite stringent measures to make sure that it's correct science. And those are peer review. Before a scientific paper is even published, it is sent to a few experts in the field. Those experts examine the paper critically to check. They want to check the validity of the method. Are there enough controls? Are there enough controlled variables? Is there proper use of statistics? Have they chosen the right statistical test? Once it passes the peer review stage, it will then be published in a scientific journal, something like Nature, something like Science. Those two that I've mentioned are quite reputable scientific journals, but there are also journals that do not have that respect yet. And actually there are some journals that do not even require peer review, and you never want to read those because you know that the science could be quite bad. The scientific community is made aware of the findings because they'll be reading the journal, and they can keep on discussing the validity of the research further. The final step is that the scientists in question will go on to present at scientific conferences where they can reach larger groups of scientists. The writer that can be questioned by the fellow scientists, whether that's the method or the results or the conclusion. It ensures that the author can justify their findings, and then it makes sure that new ideas and new lines of research can be discussed. So if it was a really specific bit of research, someone might get an idea and that research can be continued on. Okay, so your task for today is I'm going to give you an exam question on trypsin. Trypsin is an enzyme found in many groups of living organisms. Trypsin specifically acts as on a polypeptide to form amino acids. Okay, so we're going from a polypeptide chain to amino acids. State the type of chemical reaction catalyzed by trypsin. Then B, the primary structures of trypsin molecules from different species have been used to produce a phylogenetic tree. And this phylogenetic tree just shows how related organisms are and when they last shared a common ancestor. Each branch of the following phylogenetic tree represents trypsin from a different species. Your question is, Explain how the primary structure of trypsin molecules can be used to produce a phylogenetic tree. So how can we use primary structure to figure out how related organisms are? And the final questions. Trypsin molecules from vertebrates, animals with backbones, but not other animals, have a calcium ion binding site. Okay, remember trypsin's an enzyme and we're finding out that they have a binding site for calcium. Explain how this calcium ion binding site would have evolved in vertebrates. Okay, so again, I'm checking whether you understand the process of evolution. Do not get discouraged by the fact that it's for trypsin, that it's for a particular enzyme. The story is still the same as it would be for evolution, and there are 3 marks here. What I would like you to do is upload the answer, or either on Moodle, I'll make a submission link, or you can email it to me as well by 4:30 today, please. Thank you.